engage in conversation with eminent faculty from IIT Madras and other international researchers, and also to find more collaboration in those research areas. So why wait? Click the link below to be a part of this.
There should be somebody who is sort of a caring student who reaches out to every one of those students. That's how Mitra was born. Given that a student at the age of 17 joins us and matures to be an adult at the age of 21, 22,
Thank you, Shivram. Thanks for the lovely videos. IIT Madras it's a big ocean. It's very good to brush up your knowledge in daily. So, hello and good evening to everyone. Thank you for joining us today. This is Rudra from the Office of Global Engagement, and it is my pleasure to welcome you to the 51st webinar from the IRIS webinar series. So, today from the Microscopy cluster, the research initiative presenting today is Correlative Microscopy, led by Professor K.G. Pradeep. The speaker for today's program are Professor K.G. Pradeep and Professor Panikumar. So to introduce Dr. K.G. Pradeep, Professor K.G. Pradeep is currently an assistant professor in the Department of Metrological and Materials Engineering at IIT Madras. He completed his PhD from Max Planck Institute for Eisenforschung and AWT Aachen University, Germany. Prior to joining IIT Madras, he was the group leader for Autumn Pro Tomography at AWT Aachen University. His expertise lies in the fields of correlative microscopy and combinatorial alloy design. He is currently the coordinator of the National Facility for Autumn Probe Tomography, as well as the head of Max Planck India Partner Group. Joining as one of the speakers of today's program, we also have Professor Gandham Panikumar. Professor Panikumar obtained his PhD from the Indian Institute of Science, Bangalore, on the topic of experimental and computational studies on laser processing of dissimilar metals. He was an Alexander von Humboldt Fellowship at the German Aerospace Center since 2005. He is a faculty member at IIT Madras. His research interests include the integrated computational materials engineering approach. We also have Dr. Andras Kovacs, who has joined as the moderator of today's program. So Dr. Andras is a research staff scientist in the Ernst Ruska Center for Microscopy and Spectroscopy with Electrons at a research center, Hewlitz, Germany. He received his master's degree in the engineering physics in 1997 and a doctoral degree in physics, material science in 2005. His scientific career started as a research associate at the Research Institute for Technical Physics and Material Science, Budapest, Hungary. He was a postdoctoral scientist at Osaka University, Japan, at the University of Oxford, UK, and the Technical University of Denmark. His research interest focuses on studies of magnetism and magnetic materials using advanced electron microscopy techniques. So it's a great honor in inviting you all to this program. So before we start, a note to the participants, please use Q&A box to enter your question, and please like or upvote the question that may interest you so that the moderator can prioritize them. Over to you, Professor Pradeep.
Yeah, thank you, Rudra, for your kind introduction. So I hope my slides are visible. So good evening to all of you. So we would, as a team, we would like to invite you to look into what is called correlative microscopy. This is a center of excellence funded by the Institute of Eminence Initiative of the Government of India, and we were very lucky to have a very interesting team, and which came together with this idea of correlative microscopy, which is one of the most advanced and the emerging microscopy techniques across the world. So our primary activity of this center is to deal with materials design and support the materials design as well as the microstructure investigation using computational approaches, and correlate or validate all these efforts together with advanced microscopy tools, including the tool that we envisage to develop or build at IIT Madras, which I'll show briefly. So just to start with, if we have a component that something that we can see by our visual eyes, and there are a lot of these parts that are being intricate to all of these metallic structures. Now, if you want to get into the details of what could be the behavior of these materials, that can be of different metals and that can be different combination of systems. We need to look into their microstructures. So the first step that we can do is to look into an optical microscope and see in a micrometer range. That means 100 times better magnification than what our eyes can see. What could be their arrangement? That is something that is visible as a function of processing. Now, if you want to get further higher information at a much more detail, then we need to go into 100 times more magnification than what you see in an optical microscope, just with white light. We can now use electrons as a probe to look into the finer scale structures, including the grain orientations that we may see in most crystalline objects. Now, if you further want to get to further detail, that means we need to go more higher in resolution, then we can use a transmission electron microscope and then look at the nanometer scale features, including nanograins, and even at the advanced level, we can see the atomic columns even in a highly magnified situation. So we can further do diffraction studies if it's a crystalline material. We can also do other kinds of chemical composition analysis at very high magnifications of something in the range of 10 power 5 times. Now, if we need to know even higher, that means we need to go to atomic scale. Then we have these latest advancements of atom probe tomography, where we can see individual atoms visualized with some limitation in terms of the atoms being detected, whereas most of the elements in the periodic table can be identified and look for their isolated distribution or a clustered distribution. So with that information, we can further do computation studies based on the composition analysis as well as the distribution of atoms in three-dimensional space. We'll be able to do atomic scale computation till a macro scale computation. But in between, there is one missing link between these two techniques. So that means this technique enables us to look everything in a 2D projection, and this technique enables us to see in a three-dimensional form, including their chemical identity, but the real atomic positions may not be obtained from this atom probe tomography. So now the Center of Excellence in Correlative Microscopy visualizes to cover all these aspects under one umbrella, at the same time build a microscope, which is called a field ion microscope, which typically gives an image of this type as what you see here, which is million times magnified, and each individual atoms and their atomic positions are being resolved. So basically what you see here, all these greenish white dots are individual atoms, and the arrangement of atoms in a crystalline form is reflected in the form of these crystallographic poles. So using this information, coupling with what is existing as atom probe tomography, we may be in a position to locate all the atoms and chemically identify them, which is then can be used for computation, or after we do atomic scale computation, we'll come back and validate these theoretical findings from the experimental setup. So to do this wholesome business with the targets of these three fields, we need dedicated team. So the first team that we want to have is a materials design team, which Professor Nirmala, Professor Jayakanthan, and Professor Anbarsu, they will be designing different types of materials at different length scales, where all these different techniques will come in handy. So Professor Nirmala from the Department of Physics works on magnetic oxide materials. Professor Jayakanthan from the Department of Engineering Design works on additive manufactured systems. And Professor Anbarsu from an electrical engineering and physics departments works on thin film systems. So to support the materials design aspects in different forms of materials, we also have computational support in our team, and Dr. Ilax from the Applied Mechanics department deals with atomic scale computation. Dr. Narasimhan from the Mechanical Engineering department deals with molecular dynamics and middle level or mesoscale simulations. Then we have Professor Fani Kumar from the Department of Metallurgical Materials Engineering, deals with solidification and phase field phenomena. So the part of computation as we go along, we will cover in the later part of the talk, where Professor Fani Kumar and Professor Narasimhan will be dealing the techniques that are required to be used to couple all these systems together. So as we said, we are interested also to build this microscope which can reveal atoms, which is one of the very first initiatives in India. So this means we need a dedicated instrumentation team. So where I'll be joining hands together, myself, Dr. Pradeep, will be joining hands together with Professor G. Venkatratnam, who has a lot of experience in cryogenic systems, because the entire system works at, as you see here, 20 to 60 Kelvin. So we need dedicated setup and dedicated cryo stage for the atoms to be imaged as it is. So then we have Professor Sarathi from the Electrical Engineering Department, who supports us with this experience in the field of transformers and power supply systems, so that we need to apply high potential to these sharp needles to get this kind of atoms being imaged. So with this dedicated team, we intend to have a comprehensive support setup, which can first of all build such a microscope and utilize this to investigate materials of very different types produced by different people. And we will be able to have a comprehensive understanding of the materials properties, further supported by our computational colleagues. So to move on, to moderate our entire session for today, we have the support from Dr. Andras Kovacs, who is the moderator, who is from Forschungszentrum Jülich in Germany. We had a long-standing collaboration with him, and his expertise also lies in electron microscopy, where they have some of the most advanced and exciting research being going on. And they are also on the path of building a unique microscope, which he will be explaining at the later stage. So basically, I'll introduce briefly myself. So I'm assistant professor in the Department of Metallurgical Materials Engineering. So my research field deals with combinatorial alloy design of different materials, including multi-component systems and correlative microscopy. So correlative microscopy, in our case, we deal with microscopic structures where we intend to have different techniques looking into the same region of interest in a transmission geometry or in a diffraction geometry. So this is a new technique that we have developed where we can map large-scale micrometer scale structures with high resolution. So you can see these nanometer scale precipitates looking in a different matrix of grain orientations. So these things we can do for all these metallic systems as listed here. And then we can now utilize the same method to identify the phase mapping when there is a change in crystallography. Now, having seen these little pockets, if you want to see the atomic distribution, we also use what is called this atom probe tomography, so that we can identify these atomic locations and their distribution in 3 dimensions. So my colleague, Professor Nirmala from the Department of Physics, she deals with the magnetic properties of materials, where they do a lot of rare earth substituted oxide, rare earth oxides, and they produce it in the form of nanometer structures, as what you see here. There are some nanofibers and nanowires, and we support through this correlative microscopy center to do the chemical mapping and couple that with the magnetic properties of the materials at the nanometer scale. So likewise, we have different colleagues doing a whole set of activities. So we will try to cover most of them as we go along. So now to briefly introduce why this center is important, because in most of the times we do imaging, which is only a 2D projection of a 3D object. So just to highlight what is tomography, if you have a box like this, where there are subsets which can be cylindrical or which can be spherical, if you project or if you view by your eyes or by any other source like electrons or any other system, the projected image is going to be one image of this kind, where the cylinder is going to look like a rectangle, sphere is going to look like a circle, and then you will see a triangle like this. So actually, we may not know what is their extension on the third direction. So one way of approaching this problem is that if you can make multiple slices of this box and image each slices independently or individually, then we'll be able to identify reasonably their structures, but it depends on how fine we are able to slice them. So if you now combine all of this, we may be able to reconstruct this 3-dimensional object again. But this is not the case that we can do all the time at a nanometer scale. So therefore, there is a whole range of techniques that are available these days, starting from X-ray microscopy and tomography to deal with a large field of view type of structures in the range of millimeters, but a limited resolution of 100 micron or lower, coming down to electron-based tomography, and then we reach this finest resolution where you can see individual atoms, of course, with a compromise on the field of view. So we can use an atom probe tomography to see real atoms, but the field of view can be slightly extended until the micrometer. So this means we may be able to resolve atoms in materials with a limited field of view, but what we need is their accurate positioning. So if we now go into the micrometer scale, that means we can use electron microscopes to look into large field of views, and this is a thin film structure where you can now see there are different grain orientations. We can now map them chemically, we can magnify them and see there are different types of defects present inside. So that means we have the grain boundaries which are 2D defects, and then we can also have the dislocation accumulation as a function of thin film growth, which is common in most of these coatings and other systems. So when we have these kind of heterogeneous microstructure in terms of the defects present inside, there is a high possibility that certain elements which are part of the system can be accumulated at the defect sites and making them change their properties. So the interest, therefore, is to identify the different types of the defects according to the length scale available and connect it with their chemistry, which can be segregated or which can be locally positioned along these defect sites, as what we see here. So in this case, silicon goes nicely around these defect sites. Now this is still a 2D projection of all that is a 3D object. So now we are interested to see what happens to these kind of segregations if we really look them in a 3 dimension. So if you go to the atomic scale using atom probe tomography, the same silicon segregation, now you can see there are individual silicon atoms and there as well as accumulation of silicon atoms in certain specific positions looking in the form of precipitates. So this technique therefore allows us to see the atoms and their accumulation and also enables us to measure their composition accurately by going through these individual precipitates and looking into their stoichiometry. So one can therefore identify the varying stoichiometry precipitates. They may be looking similar in morphology, but they have very different chemistry. So this enables us to discern, even in a nanometer scale, variations among different local structures. So if we now continue with the atomic scale, now this is an arrangement of one of those diamond structures in one specific orientation. If we now really want to see the atomic positioning, so then we can go into high resolution microscopy, and then we can try to look at the same type of atomic arrangement at very high resolutions. And you can see this hexagon is nicely visualized. And there are locations where this atomic arrangement has a missing atom. That means open bond here. Now the question is, does this missing atom be part of the material or is it something else? That means you can also have defects exactly one atom missing from a atomic or 3-dimensional lattice. That means you have defects even at the atomic scale. This means now we need to know which atom that is missing from this precise location so that we'll be able to connect with their properties. So this is an emerging field in terms of quantum materials where diamond is being seen as a very useful material for quantum applications. So if you now really want to see this atom that is missing, then we need a 3-dimensional technique. Again, we can go back and see how these atoms are distributed and how locally they are going to change when they are accumulated around these defect sites. And then you can further zoom in and see how they are going to look like in 3 dimensions around the defect sites. So this is an example of how these nitrogen accumulates in a defect like a 2D or a 1D defect. And then you can have these individual atoms of nitrogen being also visualized. So chemically, we will be able to map everything these days and also look at the atomic scale concentration if we really go into the atomic structures. So therefore, the idea of correlative microscopy is to have a bulk material like this visualized in a micrometer scale. So if you want a specific defect region, then we can take it to an electron column where you can see the bulk sample. If you want even finer details, you can make a very thin sample out of this using a dual beam focus ion beam and couple their chemistry with and dispersive spectroscopy. And even with modern techniques, you can use the same microscope where the electron beam passes through the thin region of the sample, something in the range of 100 nanometers, going through the detector, and then you can have a transmitted beam and different types of imaging possible. So if we can now take the same imaging in a broader field of view to see the atomic layers, then you can go to a transmission electron microscope and then you can see these individual atomic columns nicely visualized. But if you now want to get exactly the 3-dimensional distribution along with the precise chemical identification, then these 3-dimensional structures is difficult using this projection methods. So you can take the same sample, convert in the form of this needle of 50 nanometer, use a detector, and then try to get their atomic evaporation being detected. And then you can see these atomic structures much more precisely in this form. So that means you can now sequentially image atomic layer by atomic layer, and you convert them, the detector signals, in the form of crystallographic holes, and you can sequentially add these as a function of time so that you get a tomographic reconstruction of 3-dimensional atomic positions as it was in the original material. So this, therefore, the same structure can go into atom probe to get the chemistry. Otherwise, we will enter into a field of atom probe field ion microscopy, and this field ion microscopy imaging is what we want to do with the new microscope that we envisage. So that means we have defects in microstructures of all type of materials from 0 to 3 dimensions, individual atoms missing, you have these individual line defects, then you can have grain boundaries and interfaces, even the 3-dimensional precipitates as what we have seen. And therefore, we are now into the process of building such a microscope so that we can resolve these atoms one by one using this atom resolving microscope to be built at IIT Madras. So to support this whole activity, we already have a set of microscopes which are extremely advanced. This is IIT Madras is one of the first institutes to have such a national facility where we have this atom probe to support us in identification of chemistry at atomic scale. We have a dual beam microscope to look at these microscale features and prepare samples suitable for atom probe. We can make it in the form of thin samples and take it to a TM, which is a transmission electron microscope, and see at much finer resolution compared to a scanning electron microscope. And the specific features, we can then take it into an atom probe and get their chemistry. So the final addition that we are going to do is going to add this field arm microscope to this entire set. So every detail from a micrometer scale to an atomic scale can be covered in one umbrella. So for more details of the national facility, you can look into this website of NFAPT at .iatm.ac.in. So our center therefore follows this work plan. So we have a dedicated team working on the material synthesis aspect of different types. Then we are also able to do imaging at this moment with the existing infrastructure. The new infrastructure is this field arm microscope that we are going to build with the instrumentation team. And at the same time, we have computational colleagues that are supporting us in this activity of both materials design as well as the post-processing of the images that we are going to get with this field arm microscope. So this again will be integrated with the national facility. So it's going to be open for anyone across India. So to say why this is important, because this is what we can get already, where you can see the atoms hitting the detector in an atom probe. We can project it together with the stereographic projection and look at their crystallographic poles together with the chemistry to a reasonable extent. But the accurate individual atomic position is not possible right now. And this is what we are going to build so that we can feed off this information to our computational colleagues who can take it from there and produce more sophisticated calculations, which is guided by experiments, or give us the output computationally so that it can be used for validating our experiments. So to explain about the computational aspects, I request Professor Fanikumar to take over and explain the details.
Thank you, Pradeep. Yeah, you can go to the next slide. Yeah, so let me first introduce myself. I would be interested in integrated computational materials engineering because it is actually very attractive in bringing together the multiple scales of microstructural simulations as well as the multiple scales at which the microstructure is being characterized. And the integrated word actually is also to bring together the various aspects that go into the engineering of a material for an end use and trying to do it in a shorter time. So in our group, we have computational alloy design being performed by talking to the thermodynamic databases and using the criteria for defect formation so that we can avoid those kind of compositions, as well as doing process simulations so that the compositional gradients, the thermal gradients, cooling rates that are actually playing a part in those process conditions can then be integrated. And we have done a couple of high entropy eutectic alloys through this kind of a design in a very short period of time. And the solidification has been my interest for several years, and we have been studying high entropy alloys and their solidification behavior. And naturally, solidification is a step that is also in an emerging manufacturing technique called additive manufacturing or 3D printing, as it is popularly called. And we basically are doing phase field simulations to bring the microstructures from our calculations so that the microstructure is no longer a piece of art that the metallurgists tend to admire, but actually a piece of sophisticated calculations that can be performed on high performance computers so that we can actually predict what kind of microstructure can come. Now, as you can see from the dendrite microstructure that is there, the iron segregation and the tantalum segregation out of the dendrite. So these kind of features today can be calculated very accurately, and thanks to also the thermodynamic databases that are there to talk to. And you can also see in the image the eutectic microstructures, which are very good because they avoid crack formation during 3D printing. So this kind of alloy development goes hand in hand with microstructural characterization at a deep scale so that we understand the materials development thoroughly. I am also interested in high performance computing to make sure that these calculations can be performed in a realistic time scale. So we do use the graphic processing capabilities to accelerate these computations. Next slide, Pradeep. So in our team, we have Ilaksh Adlakha from Applied Mechanics department, and he has been working in a range of computational techniques, starting from a macro scale where the finite element analysis is done to at an atomic scale where ab initio calculations are done. So the entire gamut of the computational techniques is explored by him. And as you can see that in the upcoming years, hydrogen is going to play a very big role because of the energy usage. And so the role hydrogen plays in how the engineered materials can withstand the defects that form and under deformation conditions, etc., is going to be important. So Ilaksh would be throwing light on the effects of hydrogen on the strength of materials by understanding how it would imply on the plastic behavior of these materials. So the microstructures that actually will contain these defect information will be shown through the characterization tools that we have at multiple scales, and also will be calculated in multiple simulation techniques that we are using across length scales, and thereby bringing a very good correlation between both the system scale all the way up to the atomic scale of the materials. Next, we have in our team Professor Narasimhan Swaminathan, who has a several year experience on molecular dynamic simulations. So he has performed these not only on metallic materials, but also on polymeric materials. And he's also been employing finite element method, particularly in the recent times on applying it to the phase field technique for microstructure evolution. And during his doctoral studies and later on, he has worked on radiation damage in materials and also on lithium ion batteries. So we have today expertise to address the defect formation in materials as well as the challenges that the energy sector is facing. Now, molecular dynamic simulations are performed at length scales that are slightly larger than the atomic length scale, but the defects that are actually seen in materials can be shown and their dynamics can be calculated quite well through these simulations. So next slide. So what we would like to propose from the computational group in this center is our capability to model the microstructure evolutions at multiple scales. So at the macro scale, where there is a component, and when you look at the component closely, you would see that there is a certain microstructure that is there, a grain structure, etc. And then we come down to mesoscale, where you have the information at the level of tens of microns, and that is where the metallurgists are fascinated by the microstructural information there. And we can go further down to microscale. We have further information, phase information, the segregation aspects, etc., and also the atomic arrangements, and all the way down to atomic scale, where the individual atoms and their interactions with the electronic cloud that is surrounding them playing a role in the defect formation at that scale. So all these length scales can be now treated in tandem thanks to the advanced techniques that can be used for computation, and also the high performance computing capability that is available in our institute as well as under the National Supercomputing Mission. Next slide, Pradeep. So I request now Professor Narasimhan Swaminathan to say a few more words about the molecular dynamics and related techniques.
Good evening, everybody. Thank you very much, Professor Fani, for the introduction on what we are planning to do in the computational part for this correlative microscopy center. So like what you all must have seen before, the understanding the role of atoms in all of these materials is extremely important. So I just want to give two examples as to how molecular dynamic simulations can actually be useful. So firstly, like what Professor Fani mentioned, radiation damage in materials happens at multiple length and time scales. For example, in the top portion, you can see point defects being generated, which is at extremely small scale, and these point defects can lead to dislocations, which in turn can actually affect the interaction of point defects with several grain boundaries in the material. And finally, the accumulation of these defects can lead to failure of the component by the formation of cracks. So there is a whole range of length scales that you can see. And in the bottom, you see various time scales that are associated with radiation damage. So when you talk about radiation damage, it is essentially an ion or a neutron coming and hitting the material and generating a large number of point defects. So this typically happens in few tens of picoseconds. After that, what happens is that the defects basically accumulate and create more damage to the material. So what we are planning to address here is the primary damage aspect, which takes place over tens of picoseconds, and molecular dynamic simulations is perfectly amenable to carry out such simulations. Can you please go on to the next slide? So here we are giving you an example as to how this is done, and we have taken the fusion materials as an example. So the breeding of neutron is extremely important in order to make sure that a fusion reaction happens. So when deuterium and tritium fuse, they generate a neutron, and these neutrons are expected to hit other atoms in the material to generate a large number of point effects. A breeder material is one which is capable of generating these tritium. So lithium is used, lithium-based are used for this purpose, and therefore they are called as breeders. So we conduct a large number of molecular dynamic simulations to understand how atoms moving in a simulation will actually hit or collide with other atoms and generate a large number of point effects. See, the right bottom plot essentially tells you how various point effects in a material have actually evolved with time over a period of about 5 picoseconds. It is this sort of information that is essentially useful in order to predict how these defects will accumulate over longer time scales and then affect the material. Can you please go on to the next slide? Yeah, another aspect that we are looking at is understanding how grain boundaries that are present in cathode materials for lithium-ion batteries are going to be playing an important role. So for instance, it is not clearly known how lithium atoms diffuse near grain boundaries of cathode materials, to what extent they expand the material, or how they actually affect the thermomechanical properties of the cathode material. So such atomic level information as to what exactly the lithium atom is doing or how they are interacting with the grain boundaries can only be obtained through atomistic scale simulations. And thanks to the new technique of atom probe tomography, we hope that we will be able to interact with some of the experimentalists in this group in order to understand these things a little bit more better. Thank you very much. Over to Dr. Kovacs.
Thank you very much. So it is my pleasure to join you here today and moderate the question and answer sessions later. So what my background is physics, material science, and thin film physics. And so what I work on is advanced transmission electron microscopy. So we study the structure and chemical properties of the material with materials with the transmission electron microscopy. And many times, even we are using advanced techniques, we have to do some to combine our measurements with other techniques, and that's why there's a correlative technique. Correlative microscopy is very important. So on the right side you see one example. So this is a samarium cobalt permanent magnet where we applied advanced transmission electron microscopy to study the structure and chemical measurements of the species, but always the TM measurements are the projection in the 2D. And this is where the atom probe tomography will be very useful, because atom probe tomography is explaining us how is the materials distributed in 3D. In addition, in the TEM, you can also measure the magnetic properties by Lorentz microscopy or electron holography. And this is electron holography can be used to map the magnetic inductions inside of the magnet, and in here we are mapping the magnetic fields of the sample. But what is very important also with the experimental technique is combined this with micromagnetic simulations. And if you do all of these together, you are very close to understand what is happening with the materials. So we can go to the next slide, which is what I would like to report, one very exciting project what we are in the middle, and we are under constructions. So this is our TOMO project, which is basically you do transmission electron microscopy and atom probe tomography, usually separately, maybe in the 2 different rooms. What we would like to do, we have built these 2 systems together. So this will take many years, and this will be a combined aberration corrected TEM, which are capable to map the atomic positions in the material, but apply these techniques on atom probe needle. So while basically we are going to find out all the species in the nanometric size samples, combining the atom probe tomography experiment with all the benefits of the TM measurements together. So I think this was all. We can go back to Pradeep with the final slides.
Yeah, thank you, Andras, for the quick overview. So to conclude from our center, so we, as we said before, we envisage to build this kind of atom resolving field and microscope and combine the same thing with the national facility so that it can be, first of all, accessible to everybody across the country. And at the same time, the information that we get from this microscope is straightforward correlation with the already existing atom probe tomography so that we can identify the atomic positions from the field and microscope, combine it with the atom probe tomography. That means this is the image we may get from this kind of a field and microscope. All these white dots are individual atoms. Their arrangement in a crystalline material is going to look with these crystallographic poles. And therefore, we can combine in a sequence of imaging like this in a three-dimensional way together with this chemical information that we can get in an atom probe in a correlative manner. So with that, we will conclude our talk. And first of all, we thank everyone involved in this project to get this kind of a unique setup being built at IIT Madras. And at the same time, we thank the funding from the Ministry of Education to support this Institute of Eminence. Through that, we got the Center of Excellence. So all these things happens quite well so that we can make this kind of bold initiatives to build microscopes in the country. So I thank Andras for being patient to us and gladly accepting our invitation to moderate our webinar on this new session, and thank all the participants for listening to us. Thank you. So Andras,
Thank you very much for the presentations. I've seen many interesting results and future directions in the slides presented in the last few minutes. And we already have a couple of questions in the chat box. So this is I will going to read. And one of the questions, Shital Kavala is asking about the plastic nature of the material. So I think this is a very good question because we understand the materials, but functional properties of the materials is very important. So probably Pradeep can comment. So how is the plastic nature or plasticity can be studied in an SEM, for example?
Yeah. So together with the center, when we have our dual beam microscope to prepare this kind of a sharp needle, we also intend to have a deformation platform. That means we bring a material which is in a homogeneous state, apply certain levels of deformation so that you can induce or generate defects when the material is plastic and crystalline. That means you enter into a zone where you induce permanent deformation, and the regions of interest, for example, dislocation or the slip system active, being investigated by these techniques. So the method what we are developing is not restricted to only imaging. So the stage what we see here, it will also be carrying a mechanical deformation platform so that we can do these plasticity experiments and at the same time use these samples to produce these kind of sharp needles. And then we can investigate again by imaging them, and then we can correlate with their chemistry. So basically we have this add-on capability to be added to this infrastructure. So we can then do the plastic deformation at room temperature as well as at elevated temperatures.
Yes,
yeah, I hope that answers.
I think so. You and I understood. So I think regarding we probably with the next question we are staying with an SEM because Sarah Chandran is asking about a slice and view technique for the 3D projection. I think this is a valid question because atom probe tomography typically gives you the reconstruction of the 3D of the atom positions in 3D of a few cube nanometer space. And that's why it's probably the slice and view, understanding of the slice and view is very important. So please explain.
So the system that we have is a dual beam microscope. So it's not just that we try to image them. So probably I'll see if we can go back to this. So we can even look into these materials in the cross section in all three directions. So that means we have this dual beam microscope where you can get this kind of a surface imaging, and then you can have this focus on beam as I just showed in the earlier schematic as what you see here. So the bulk surface can be imaged, then we can do a serial cross-sectioning across the surfaces of the regions of interest using this focused ion beam. That means for each slicing that we do, we start to see their cross-sectional microstructure also. So we can rotate the samples, we can tilt the samples in any direction as it requires, and look through their three-dimensional sectioned image. But we have to remember that each sectioning that we are doing is a destructive method. That means that particular region, what was originally present, would have been lost. So if we do a serial section imaging like this, even in a bulk scale, using the existing infrastructure, we'll already be able to get a three-dimensional sequential imaging, which will then be reconstructed to look like this. So this is more or less in the micrometer range scale. We can extend this to a nanometer scale, like what you see here. We have a thin section being taken out of this bulk and then image it with this technique. So that means you have electron transparency by producing these thin sections from a bulk sample using this focused ion beam. And then those thin sections can be imaged both in a transmission mode in a scanning electron microscope or in a transmission electron microscope. And even if right now we don't do it, but there are places where you can again tilt these thin samples and get images at different directions and try to combine them into a 3D information so that you don't miss out on some of the features that can be visualized only in the 3D. So therefore, we have, at this moment, we have all the capability to do it in a bulk scale, in a micrometer scale, with cross-sectioning by selective field milling and look through the third dimension also by imaging. And if required, we can go to the nanometer scale and look them. And further, we can go to the almost atomic scale by imaging using these atom probe tomography methods.
Indeed. Pradeep, I have a question actually. So because we went through half of the periodic table basically, so what are the elements can be detected, but how about the very, very light elements to detect? Because if we are talking about hydrogen, so hydrogen in pretty much not possible to detect with transmission electron microscopy, and basically the background of that, so the electron wave, not really interacting much with the hydrogen atoms or molecules. So how about the atom probe tomography reconstruction? So can you comment on that?
Yeah, very good question, Andra. So this is a interesting challenge that is facing atom probe for a long time. So whenever people wanted to study hydrogen-induced effects like embrittlement in steel or other materials, so the easiest way to deal right now is charging deuterium instead of hydrogen. And then the charged deuterium, if we can retain in the material, probably at the defect sites being accumulated with this deuterium, and those species of deuterium we can detect. So in principle, even when the atom probe runs at ultra high vacuum, the hydrogen is very difficult to be eliminated. So even in a natural measurement where we do realistic samples, we see traces of hydrogen being detected, but we will not be able to say this is from the material or this is from the ultra high vacuum chamber. But if we now selectively add deuterium in defined proportions into the sample like this, and that deuterium can be well identified, and since we already knew how much we might have charged, we'll be able to look into their quantified data, whether this equivalence of deuterium is available. Then one can definitely say that the deuterium that we charged is really coming from the material, and we will be able to identify their positions in 3 dimensions. So one can say if deuterium is really going to the defect site or it is just dissolved in the matrix. So at least we can get a first-hand fingerprint of what is this deuterium doing, but not exactly hydrogen, but of course is the same family. So we can say that we'll be able to understand a little bit better than not having the information. But the interesting challenge what we see, for example, the samarium system that you have shown earlier, we have noticed that samarium quickly reacts with hydrogen and forms samarium hydride. And some of these diffraction, what we have been doing in the SEM, seems to be a challenge. So we are unable to get diffraction patterns when we work with elements like samarium, which seems to have very strong affinity with hydrogen. So those samples may be tricky, even if you want to study about the hydrogen effects in their materials. But for most steels and superalloys, I think charging hydrogen with deuterium solves the problem.
Yes, thank you very much. It's very nicely explained, and I think you also mentioning the interaction of samarium with hydrogen is important because we indeed use the hydrogen treatment in many material science processes. So probably the next, we received an excellent question. So maybe we can jump from hydrogen to carbon and maybe carbohydrates, because Scientica Sanyal is asking about how the correlative microscopy can analyze different folds of protein structures on atomic scales. And I like these questions very much because so far we've shown examples for material science, but I think atom probe tomography and electron microscopy we know very well. So biology and structural biology is used very extensively, microscopy techniques to analyze biological samples, but indeed the proteins, maybe the proteins as well. So can you comment how is the protein structures and what are the other applications other than material science for atom probe tomography?
Yeah, so we have just started exploring protein structures from one of the projects. We have not yet matured in that state, but what I can say is that it should be definitely possible because we are able to work in atom probe at cryogenic conditions, and we are trying to evaporate species one by one. So in principle, it should be possible. But what I can say is that if a material like diamond, where there is full of carbon here, if this can be imaged without significant damage in an electron microscope at high resolutions, in principle, protein structures, as we know, even with cryo-electron microscopy, we should be able to image them nicely. But when we take it to atom probe, this interim period where we prepare these samples in the form of a needle rather than an electron transparent lamella, this is going to be the challenge. As long as we are able to preserve those structures without damage or decomposition of any other form of a beam being interacting with them, we should be able to get this kind of arrangement of whatever atoms that is made of inside, and we should be able to map them. So only question is, it is possible that some of these segments are strongly bonded in proteins compared to the metallic systems. So we may get chunks of these elements being identified in the 3 dimensions. But I think over a period of time, we should be able to overcome this with the optimization of the laser that is being utilized for the process of evaporation in atom probe. And at the same time, by further advancing our sample preparation techniques using the cryo stage in a focus ion beam, we should be able to produce samples which are highly stable and transfer it under a cryo condition into the atom probe so that we are able to preserve those samples from significantly decomposing, let's say, or any other form of damage that is being incurred on the sample. But there are groups that have started exploring the biological materials in using atom probe. And we believe that in the first phase, if we are able to get this field and microscope working, we should be able to integrate it at some point with the atom probe. So with the possibly a cryo transfer facility, this will enable us to explore protein structures in other biological systems.
Yes, so this is probably also the answer to the question of Monica Singh, because she was asking about the correlative microscopy of living cells. And probably I can comment. And so living cells, as it is probably not possible because all of these biological samples, first of all, have to be freezed. So the cells are embedded in vitreous ice. But after that, as you very nicely explained, if you can maintain the transfer in the cryogenic conditions to an atom probe and we don't destroy before, so the experiment can be successful. So I can foresee more and more applications of cells and biological applications also at an atom probe. We have a few questions from Roshan Vani about whether correlative microscopy can be used in space research. So that's an interesting question because, well, indirectly, so electron microscopy, what we are doing is an optical technique, and the Hubble telescope and other things is also optics, what we are using to reveal the secrets of the space and the universe. But I think here we study the material science of materials what we are preparing, and this probably cannot be used directly for the space research. But I would like to comment indirectly, yes, because we have a lot of material on Earth, like in the form of meteorites, and meteorites coming from the solar systems. And in this way, we can study the compositions, for example, what is happening on the other planets, because in the past, if a big meteorite hit the Mars, other fragments are arriving to the Earth in the form of meteorites. In this way, we can pick up a stone in the earth and study it. And in this way, we study what is the composition of the minerals in Mars, different planets. So in some way, indirectly, I would say, yes, it is possible also to study in the space research. Pradeep, the next question is because we received one question for the field ion microscopy, whether this can be used, how to characterize nanomaterials. So can you please comment what is the functional proper, what is the field ion microscopy to be used for? So how is maybe complement to the atom probe tomography?
Yeah, good. So as what you see here, so this schematic nicely explains that when we have these materials or atomic arrangement in materials being looked in with an electron beam, we get a projected view. But the same sample or a different geometry of the same thickness can be produced in this order. That means you have this periodic arrangement of atoms in most crystalline materials being arranged in this form. But if we now do atom probe tomography, we are evaporating atoms one after the other. That means it's a destructive process. We are losing material layer by layer as and when the process of field evaporation continues. Whereas in the case of a field ion microscope, we are not going to evaporate any atom. So we are going to apply a positive potential, and we are going to inject gas species. That can be helium, that can be argon, whichever that's not going to be reactive. So because we are applying positive potential, it will be strongly polarized. The atoms at the king sides or the ledges here will be strongly polarized, and the incoming gas species gets ionized by interaction at the surface. So once they are ionized, they are going to hit the phosphor screen or the detector. So wherever it's going to hit, it's going to form this fluorescence, and therefore we'll get a bright image like this. So this is what we are going to see. So the darker area is a detector when there is no atom hitting, whereas these bright spots, as what you see, these are all images of individual atoms sitting on the surface of a sharp needle like this. So that means wherever there is an atom being there and the imaging species gets polarized to that particular location, and the ionized gas species will hit the detector starting from that location is being nicely mapped here. So when the structure is fully crystalline, we'll be able to see these crystallographic poles so that we can be able to identify which orientation of the grain this tip is made of. So one can therefore see, so there will be a ring structure as what you see here, and each ring structure corresponds to one atomic plane. And at some point, if you start evaporating the species also together, we'll see that this ring structure here starts shrinking. So that means it starts losing atoms one by one as the atoms get evaporated. So the more these rings shrink and the more the rings disappear, one can even measure the number of circles that are disappearing to measure the nanocrystalline grain size. So this is an image of a nanocrystal where we have multiple different nanocrystals oriented in different directions surrounded by an amorphous matrix. So when you have an amorphous matrix, the atoms are randomly distributed, so you don't get any periodicity here. Whereas these nanocrystals are visualized through these orientation difference from their crystallographic poles. So this is easiest way of looking into the field ion microscope for nanomaterials. But if you have a micrometer scale grains, then you will see all of the crystallographic poles nicely visualized as what you see in the case of a stereographic projection.
Yeah. Thank you very much, Pradeep. So staying with the field ion microscopy, we received the questions I don't fully understand, but so it sounds like, why can't we relate field ion microscopy to any AI? And I thought AI is maybe artificial intelligence. And I was thinking, so can you comment, so how is the computational methods maybe can help you with the reconstructions? And what I mean actually, so in these days, so the machine learning and artificial intelligence and pattern recognitions is very, very hot topic, and we use more and more in our fields, in different fields of microscopy. So can you please comment how is these techniques can help us in the future or now?
Yeah, so correct. So this is again already people have started exploring this type of subject where, for example, if you get these atoms being imaged in 3 dimensions in an atom probe, it's not that every atom that is being evaporated is being detected. This is a deficiency on the physical state of the detector. So that means at some positions we may miss out on some atoms. But by doing field ion microscope, we already know the exact crystallographic orientation of the grains that are emitting these atoms. So that means these missing atoms from the detected region can be iteratively identified for their positioning using this computation approach. And so as that we can go back and see if there is sort of a real lattice reconstruction that can be built, and it can be therefore utilized for cross correlation with their chemical distribution. On the other hand, it is also possible that even though we may say the shape of the tip is going to be this 50 nanometer, the electrostatic potential that's being applicable here is over the range of few volts per nanometer. It is still not accurate prediction of what is the exact curvature of the tip and what is the exact potential that is being applied. It is just relative, and with a good guess, we are able to get reasonably good images. But a significant modeling is required over ranges of these kind of surfaces and see what is the potential existing at the surface at what distance. So this will enable us to go back and verify whether there is any distortion associated with this evaporation. So both these issue of distortion in the atomic positions with respect to the applied field and the issue of missing atoms because of a detection deficiency can to a large extent be overcome with the computational effort. The one more important aspect of computation effort is that after we do this reconstruction, most often we see a situation where atoms are being locally clustered. That means we can have, for example, a short range ordering in a large matrix. So with the existing tools, it is very difficult to identify the short range orders. If you have 3 atoms, 4 atoms forming a cluster, and 1 atom potentially missing because of this detection, we may miss out on this short range order information. So where the large data sets over different sequences of imaging would enable us to identify these short range order clusters using the computational tool, which will enable us in understanding the material's properties in a better way. So I think in all the 3 aspects, there is a large scope of utilizing the artificial intelligence tools, because there are already a lot of experimental data sets available for same type of material. And at the same time, we also have databases of the analyzed results, which is part, I mean, some of them may be published, some of them may not be published, but from the same family of materials, we have wholesome lot of data sets, which can be utilized to do this post-processing and get out much more fruitful information, which we are missing right now.
Thank you very much. So actually, this is some of the problems you mentioned about not knowing the exact shape of the tip and so on. This is one of the reasons why we are building our Tomo project, because in the Tomo project, during the characterization, you can, with holography, you can map the electrostatic field around the tip and with the materials properties as well. So you can probably have a kind of a very smart feedback. So what kind of laser power or pressure you have to apply to evaporate atoms. And well, I think we are running out of questions, and probably we are also running out of time, because already our discussion is going for 20-25 minutes. So should we
ask once our computation colleagues to add something on artificial intelligence with these atomic scale correlative techniques, so that we can, it's a good platform to say they have capabilities to do this in our center.
Yeah. Briefly, I can mention that the emergence of various algorithms and libraries to implement the machine learning, as well as the semantic aspects, have made the AI quite ready for us to take advantage of in materials, including the microstructural analysis. So our group has been working on using the machine learning to speed up the microstructural simulation, and where some amount of calculations can be postponed because the pattern recognition and prediction that is possible by algorithms like LSTM can be taken advantage of. And with regarding to the kind of calculations that Dr. Pradeep has mentioned, I must say that anytime there is any calculation that is very tedious and involves a lot of complicated equations, but a set of such calculations, if they can be made and preserved, then definitely repetitive running of those calculations is not necessary, because today the machine learning techniques have advanced a lot, and we can definitely speed up some of those aspects. So surely the techniques of AI are very much within the ambit of the computational toolset that we are planning to use in this kind of a work.
Thank you very much. And we have one question, actually, maybe it was coming very early. So Sarat Chandran was commenting how is a high resolution projection in nanoscale level can be achieved. And I think this question relates to well, maybe not to, but I think what we haven't touched today is how important the specimen preparation. So because atom probe tomography heavily rely on the focused ion beam, because all of this preparation with the very fine needles with the nanometer sizes. So I think it is amazing how is the focused ion beam technique can be used, but also for the TEM, I would like to comment. So we can buy expensive machines and do high resolution analysis, but this is not important. What is very important is actually the specimen preparation. So the fancy machines are useless if you don't do the specimen preparation properly. So probably this is maybe the final comments we can do here today before we close.
Okay, excellent. So I think we can give it back to Rudra if we are done with all the questions.
Yes.
Okay. Rudra, over to you.
Thank you, Professor Pradeep. Indeed, to comment, it was a very good program that we had today, and I can see like a lot of questions that has been asked by the very intellectual. And thank you once again, Professor. Thank you, audience. Looking forward to meeting you in the next webinar. Thank you.